Thank you everyone for joining me today. My name is Laura Miles and I'm a Senior Application Specialist at Marks International. In this session, I'm going to be taking you through how thermal desorption can be used to meet the evolving demands of clinical breath analysis and help to take the current research to the level of routine screening. So what is thermal desorption, or TD? TD is a pre-concentration technique that can be coupled with GCMS to provide the gold standard approach to taking and analysing breath samples in the discovery stage. It's a flexible sampling technique enabling the breath VOCs to be taken onto a sorbent tube directly from patients or from breath samplers, sampling bags, or dedicated sampling masks. The sorbent tubes are then capped and transported securely to the lab where they are analysed on an automated thermal desorption system with a GCMS. The combination of these techniques enables easy sample collection with separation of the wide range of compounds present in breath and identification with spectral matching or standards. One of the most important things to consider is the security and integrity of samples that are taken within this application area, so quality control at every stage from sampling, storage and analysis is imperative. Thermal desorption systems can offer a number of features that can help with this that are particularly useful throughout long-term studies such as clinical trials, including leak checking of every sample and data traceability with sample tracking. However, I'm not going to get through all of these today, so I will just focus on a couple and talk through them all in more detail. Firstly, let's look at internal standard addition to help with safeguarding sample integrity. A gas phase standard can be automatically added to tubes prior to analysis, and this can help to monitor instrument performance over time. And we can see this in the data in the graph on the left-hand side, where toluene D8 response has been used to monitor instrument performance and excellent reproducibility is observed. Any deviations from this profile would highlight that an issue needs further investigation and can highlight if any samples need to be reanalysed. As well as this, a standard can also be added to the tubes prior to shipping out for sampling in the field. In this case, we use benzene D6 as a surrogate. This should mean that all tubes analysed should have both benzene D6 and toluene D8 present to confirm the integrity of the sample. Any drops in response of these compounds, such as in sample 2 on the right-hand side, where benzene D6 is missing, this would indicate a failure in the process. In this case, a failure in the process between sampling, transport, or storage. This is a really quick and easy way to track the integrity of the samples, especially over long periods of time. So next, let's look at managing water, which, as I'm sure you know, is a big consideration when choosing a method to sample and analyse humid samples such as breath. Dry purging is a feature where water is chromatographically removed from the sample tube by flushing it with dry gas in the same direction as sampling, causing water to be purged off the end of the tube, but the VOCs to remain trapped on the sorbent. So what you can see here at the top is a chromatogram of a breath sample where dry purging was not performed. In this example, we have dropped the scan range down to include the mass charge of 18 to be able to detect water. And you can see there is a section with an elevated baseline with some small peaks present that could be target analytes. If we compare this to the same sample that was then analysed with dry purging, which you can see at the bottom, in this chromatogram you can see optimum purging is being performed. So the same peaks have a much higher response now the elevated baseline from water has been removed. This has increased the signal to noise value and therefore has improved the overall sensitivity with one easy method change, whereas in the first image the results would be underreported. If we also extract and overlay the ion for water, you can see the direct comparison of dry purging between the two samples and the effect it has on managing water without loss of compounds. Not only does this have a big effect on the results, but also as this is topping out the detector, it's likely to result in columns and filaments needing to be replaced sooner, and sources and mass spec needing to be cleaned more regularly, meaning more instrument maintenance and downtime. The last important feature of an optimal thermal desorption system that I'll be talking about today is the option to recollect samples. What this means is that when a split flow is applied during analysis of a sample, this portion of the sample can be recollected onto a clean sorbent tube, and the sample can be archived for reanalysis in future. This means none of the precious sample from patients who have volunteered their time is wasted, and provides the option to reanalyze the same sample, which is beneficial for several reasons, including validation of any future method developments, but also for peace of mind to safeguard any instrument issues, such as contamination, which would mean the result would be lost otherwise. This is extremely valuable in many applications, but particularly when you are dealing with breath samples from a specific point in time of a patient's clinical journey through various stages of treatment, where samples cannot be collected again. So what's new to help with the scaling up from biomarker discovery to full clinical trials? Samples in this area are typically complex and highly variable, so it's impractical to create a truly representative standard for breath analysis to cover everything. In other biological testing, a small aliquot of samples such as blood or saliva are taken and pooled together to create a QC that contains a representative portion of all components of samples within that analytical batch. The blended sample is then distributed throughout the batch, and during data processing, this can be seen clustered together in a principal component analysis, as you can see in red on the right-hand side. What this enables us to do is assess the data quality, measure the reproducibility, and correct for any systemic bias. To apply this using thermal desorption, Dr. Wakar Ahmed and the team at University of Manchester have used the recollection capabilities of the TD instruments that they have to create pooled QC samples. So during step A, a sample is analysed and a split portion of the sample is recollected onto a sorbent tube. This is repeated for multiple samples to generate a pooled sample with all split portions being recollected onto the same tube to create the QC. In step B, the pooled sample is analysed with the split portion recollected again, and this is performed multiple times throughout the analytical sequence. Each time this process is performed, the split flow is adjusted to maintain the same amount being sent to the column each time. By following these steps, they have demonstrated how a pooled QC can work for breath analysis. So in this example, we have breath samples from 3 participants, including a baseline test, 1 hour post-peppermint capsule, and 1 hour after having a coffee, with each collected in quadruplet. You can clearly see the separate sections in the PCA plot, where the sample post-coffee is separated out from the others on the left-hand side, with the QC sample tightly clustered in the middle between the various sample types, showing great reproducibility of the system. So this example has shown how the instrumentation can be used in this manner to create a pooled QC containing a representative VOC profile of real samples, and with good reproducibility. But what about its usefulness for longer-term studies? We have a look at the images on this slide. The top image indicates the responses, where we can see that when the instrument was tuned between batches, there was a change in response, which you would expect, but they are quite significantly different. Using the pooled QC, the drift in response can be corrected by subtracting the mean peak area of the QC from the grand mean across all samples. This is a great demonstration of how this can be used across longer-term studies, such as clinical trials, by using actual biological samples with representative profiles to perform batch correction in a field as complex as metabolomics. So to simplify this process, here's a quick preview of something we've been working on to help generate pooled QC samples prior to analysis. So looking at the image, you can see a sorbent tube is placed in the oven at the centre, and this is then heated with a gas flow to desorb it onto up to 10 other sorbent tubes. This splits the original sample, enabling replicates to be produced, whilst only one sample is required to be taken during collection and transportation. Not only can this be used for sorbent tubes, but also for desorption of other sampling media such as thin film SPME and high SOR probes that have been used for sampling skin surfaces or saliva samples. These can also be desorbed in the same manner to provide pooled QCs, but has the added benefit of enhanced stabilization of biological samples by storing them on solid sorbents, meaning they can be stored for longer periods of time and helping to provide a more complete view of a patient's history. So today's presentation was just a snapshot of solutions that Marks International can offer for dealing with the challenges of analysing breath. So please reach out to us for any support on the challenges I've just discussed or any other challenges you want to involve us in, and we would be happy to help work to resolve these with you. Thank you for taking the time to listen in, and I'll be available for any questions.
That was great. Thank you so much. It was wonderful, wonderful to see the progress. That's been made. It's very exciting.
Yeah, yeah.
So one question here is, do you have SOPs or best practice guides that you can share with the breathomics research community so they know how to deploy those?
Sorry, so SOPs for
Or best practice guides to cover some of the elements that have been discussed today.
yeah. So we have a application note available that has a lot of information on best practices for analyzing breath samples with thermal desorption. It takes you through from start to finish, all the different considerations. So you will be able to find that on our website. Yeah.
Brilliant. Next question is, are there any best practice tips for storing of breath samples? So I know dry purging matters a lot, but I wonder if you could elaborate further on storage.
Yeah, that's right. So with the for storage, the first thing we want to do is dry purge our tubes. Obviously, we want to make sure we're getting rid of some of that water because there's obviously quite a lot in breath samples, and that's going to help to stabilize any VOCs that you've got on the sorbent tubes. After you've done the dry purging, you're going to put the long-term storage caps onto the tubes and place them inside something like a non-emitting container that's sealed. And then place them inside something like a fridge or freezer to store them for the longer periods of time. The most important thing during this is to go back once your tubes have sort of equilibrated at the temperature in your freezer. To go back and retighten your caps on the tubes, because otherwise, obviously, with the metal contracting, there's going to be tiny gaps, and you don't want to compromise any of your samples there by letting in any tiny bits of gas. It'll just carry on sampling the inside of your fridge or freezer otherwise. So it's really important to go back and do that. And then the same in reverse, really. If you're going to take them back out of the freezer, let them come up to room temperature first before you change over to any analytical caps.
Brilliant, thank you. Next question relates to the recollect. How many times can you do a recollect?
That's a good question. So in theory, as many times as you like. Obviously, we're dealing with low concentration compounds. With recollection, you can do this automated within the system. So if you wanted to do something, something that's really popular actually in breath analysis is what we call a high-low recollection. So you've got an analysis of your sample at a lower split flow. So we're going to recollect that. And we're going to be looking at our trace level compounds, and then we're going to run it again with a high split flow, and we're going to be looking at our more higher concentration compounds then. So we're going to be able to calculate the concentrations of those using the split flow ratios. But then you can use your recollection multiple ways, really.
Fantastic. And the system you showed it then, where you're going from single tube and splitting on the multiple tubes, is that available now or is that still in development?
Yes, that's something we're working on at the moment. And if you're interested in knowing more, then please feel free to email us, and we can start some discussions on that. It's something that's still in progress, but we wanted to share it because we think it's really beneficial, especially in this area, especially when we're moving into looking at more of these sort of pool QC types and looking at real matrix, something that's really important.
Yeah, I think it'd be super helpful. And as you said, the clinical samples are so valuable.
Yeah.
And if you're looking to analyze with different analytical techniques, I think this would be a very valuable piece of equipment. The last question relates to the sorbent materials. So could you comment on the types of sorbents used in breath analysis? And is there any research that Marks does into novel sorbent materials?
Yeah, so the sorbents that we are looking at specifically in this area are going to be hydrophobic to make sure that we're not trapping water where possible. So typically the sorbent tubes that we recommend are the biomonitoring tubes. They are hydrophobic. They're also inert, so we're not going to run the risk of having any of our compounds sort of reacting with each other on sorbent tube after we've taken a sample. But we also want to trap the widest range of volatility, right? Because we don't know what we're looking for sometimes. If we do, typically it's quite a wide range of compounds. So that's in general the sorbents that we would recommend for this.
That's brilliant. Well, thank you so much again for presenting today. As said, sounds like some fantastic work. And we'll share your details with everyone in the audience as well. A few people have said, can we have Laura's email address? So we'll send that to everyone.
Yes, that'd be great. Thank you.
Brilliant. Thanks so much.